Welcome to Lecture 17. Like Lecture 14, this one will be split into two. I will try to stop about 50 minutes through, and then we'll resume with the B part of Lecture 17. This lecture is like 14 in that it involves several classes of compounds, however, not as many as were in Lecture 14. Again, like Lecture 14, this lecture is going to encompass several sections of the textbook. As always, these are outlined on your calendar as to which sections are covered in your text. We're going to start with proteins, which I believe is chapter 19. Carbohydrates, I think that's chapter 15, and then lipids, chapter 17. Like we did in Lecture 14, we are just going to be examining the structures of these molecules. Your knowledge of functional groups from organic chemistry, Lectures 13 and particularly 14, are going to be quite useful in understanding biochemical molecules because they are carbon-based, so they are organic in nature. First one thing we're going to talk about are proteins, and proteins are very, very important in terms of what they do for our bodies. Carbohydrates, when we get to those, are mainly energy molecules and storage. And then lipids, same thing, energy and storage, although some are also steroids and hormones. Proteins are pretty much everything else. Structural, collagen, tendons and cartilage, hair, skin, wool, nails, muscles are made out of proteins. Proteins transport things throughout the body. Hemoglobin transports oxygen and so on. Store nutrients. Not as much as carbohydrates and lipids, but casein stores protein in milk. So they're storage molecules within things. Ferritin stores iron and so on. Hormonal, very important part of protein chemistry is hormones, the chemistry of hormones. Some hormones are steroidal, which means they're lipids, but most are proteins. They regulate our metabolism and nervous system. Enzymes are all proteins, and they catalyze all biochemical reactions in the body. And then protection, your immunoglobulins are stimulate your immune system. So the thing that I want you to recognize in the name, so that when you see the name of something biochemical, something in the body, if it ends at an N, I-N, it is probably a protein. You'll see a different type of ending for carbohydrates and a different type of ending for lipids. So this N ending gives you the clue that it's a protein. Proteins are also important in DNA, in the expression of our DNA. That is done through proteins. Oh, right. Sorry about that. All right, proteins are made up of amino acids, and they are the building blocks. They can contain a carboxylic acid group. So here is your carboxylic acid group. So again, that functional group chemistry from Lecture 14, very important. Every single amino acid, the acid part comes from this carboxylate or carboxylic acid group on one end. And at the other end of the amino acid molecule, every single one, amino, there's an amine group. So again, functional group chemistry. So the basic amino acid is a carbon in the middle, an amino group on one side, and a carboxylic acid on the other. This makes it very much like an amide. However, you'll see when we start putting amino acids together that they do in fact form an amide or amide linkage. The amino acids are distinguished from one another by their R group. The middle carbon is where the R group is attached. So you have a middle carbon, which always has a hydrogen on it, unless it's attached to something else. An amine group on one side, carboxylic acid on the other side, and then an R group. This is what distinguishes the 20 amino acids from one another. So when the R group is a methyl group, that amino acid is alanine. The types of chemistry that are associated with these different organic groups is going to hold true as well. So like a methyl group is nonpolar. So we're going to be separating these 20 amino acids into whether they're polar or nonpolar, acidic, and so on. This last bullet right here, ionized in solution, you'll see on the next page what that means. So at the pH of most bodily fluids, amino acids are ionized. What this means is that this hydrogen right here on the, oops, sorry, I hate that I have to do the pen thing on every single slide, so give me a moment. This hydrogen right here is taken off and then put on the carboxylic acid group and put on the amine group. So this now you've lost an H plus, so now it has the negative charge on the carboxylic acid group, and this nitrogen with 4 bonds to it has a positive charge. You just put a plus, an H plus on it. If you remember from our discussion of bonding, this is called a coordinate covalent bond, or we got some Lewis acid-based chemistry going on here, which you'll learn about later, because we're taking an H plus that has no electrons off of the carboxylic acid group, and we're putting it on the nitrogen atom. That nitrogen is not involving any, I'm sorry, it is involving both of the electrons that are in the lone pair. That's not shown here. There's a lone pair there. I'm not getting it right. Okay. So there we go. So you've got a lone, that is really messy. You've got a lone pair on this, not letting me do it, on that nitrogen, and it's donating both of those electrons to that empty orbital in the H plus. So big discretion on a bonding there, not so important, but this ion, this ionized form of amino acid is called a Zwitter ion. It's a dipolar ion. You have a negative end and a positive end. So this is going to make the amino acid very soluble in water, all right, if it's just an amino acid. So as I said before, they're classified according to their R groups. If the R group is nonpolar, of course, it's not going to dissolve in water, so it has a hydrophobia. It has a phobia for water. If it's polar and neutral, meaning like you have a polar group, and we'll see that, you've got some oxygen and sulfurs, but there's no charge, then it's going to like water, and we call that hydrophilic. If it's polar and it's got some acid groups on it, because it contains a carboxylic, oops, because it contains the carboxyl group, remember that is COO negative. Or if I'm going to draw it out in a structural formula, I'll do it up here, it's C double bond O and then O. And this whole thing has a negative charge. All right? If it's basic, polar and basic, then it means it has amino groups. And you know what that means. It means it's NH with either 2 or probably 3 if it has the positive charge. So NH3 plus. And as we saw in the earlier slide, every amino acid has this and this if it's an amino acid by itself. And when we combine amino acids and make peptides, then those groups aren't there anymore. So we are talking about the R groups here, not about the carboxylic acid group that's present on every amino acid when it's by itself, or the amino group that's present on every amino acid when it's the singular amino acid. Okay, so nonpolar. In blue on this slide and on the slides right after, it's showing you the basic backbone, if you will, for an amino acid, carbon in the middle, amino group on one side, carboxylic acid on the other, and that hydrogen group, as long as that carbon is not involved in any other kind of interaction. Go back. So in yellow are the R groups. All right, so a nonpolar group would be something that's very similar to an alkane, so it only has carbons and hydrogens. Remember, that's nonpolar. So this is nonpolar. However, this right here with an OH group, with an alcohol group on it, is polar, but since it has no charge, it's called neutral. This R group here has the carboxylate. Remember, we're only talking about R groups, not about this one. So that makes it acidic and polar. If it's basic, it has the amino group and has a positive charge, and that is going to be polar as well. Now this group right here has a significant nonpolar part, but with that polar group at the end, it'll make this a polar amino acid. All three of these are polar, right? This is nonpolar. And that's talked about here and in the previous slide. Now, let me say just one more thing about these charges. At the pH of our bodily fluids, these groups, just like this one, have the charge. If you're at a different pH, that's not going to be true. So, if you're at a low pH, then you have lots of H pluses around, then this charge on this carboxylate ion will not be here, it'll be OH. That's at low pH, and we haven't talked about pH yet. And then if you're at high pH, you're basic, you have a lot of OH minus around, which means it's going to take away that H plus. We're talking about water-based systems, so this will not have the plus charge. So we are talking about the charges that these amino acids would have at the pH for bodily fluids. So if we have an alkyl group, think alkane. So that means nonpolar, all carbons and hydrogens. It could contain an halogen as well. If it's aromatic, then it means, like we've talked about briefly, that it has this ring, this type of ring on it. These are nonpolar. They're always going to be neutral. So here are your nonpolar amino acids out of the 20. Glycine, alanine, valine. Now, a couple things to notice. One is there's a number written under here, and that number has to do with electrophoresis. We're not going to talk about that. The abbreviations for the amino acids are important. Your book, and at least the examples in these PowerPoints, use the 3-symbol way of abbreviating amino acids. I am most familiar with the 3-symbol. That's why I was taught. However, there's also a 1-symbol abbreviation, a little harder, because there are a number of amino acids, more than one, that start with an A. So though others that start with an A don't have an A as the single letter abbreviation, I will use the 3 letters because it's just the way I was taught, and I was never really able to embrace the single letter. I will never ask you about the single letter abbreviations. And given the nature of how much biochemistry we're going to talk about, that'll be just fine. All right. So if you look at every one of these groups in the yellow boxes, you see that alkyl, 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 just a hydrogen, all of these are very nonpolar. This group right here is very aromatic and nonpolar. This sulfur group is here, so we might say polar and neutral, but it's in the middle of alkyl groups. This here is an alkyl group, and look at that funky ringy thing going on, proline. And here we've got not an aromatic, but a, well, yes, we do. We have the benzene ring right here, then we have another ring next to it, and most of this is, this molecule here is nonpolar. If this hydrogen, because it's locked up in this ring, may or may not accept another hydrogen, but we're not going to go there. Tryptophan is that amino acid. So these are your nonpolar amino acids. Your polar but neutral means that they've got polar groups on them, so you know that the presence of an OH group is going to tell you, oh, polar group at the end, oxygen, high electronegativity, OH group at the end. What is the functional group for these 2 amino acids, serine and threonine? Yes, it's an alcohol. That's the functional group for the R group on those 2 amino acids. Here we've got, what is this molecule right here called? Reviewing our organic chemistry. That is phenol. Now this whole R group is not phenol because it's got the CH2 group here, but it almost looks like I'm not going to go there. Anyway, it's polar but neutral. Here we have a sulfur group, not as polar certainly as an oxygen group, but it's at the end, unlike in the previous slide when you have the sulfur in the middle. Cysteine, very important amino acid, because this two cysteines or a cysteine in another molecule can form sulfur. They can bond together in what's called a disulfide bridge, and we'll talk about those in a minute. Asparagine and glutamine are amides. Remember, you've got an amide group. You've got a carboxylic acid. Instead of an OH, you have an amine group. So these two are amides. So knowing our organic functional groups, very important. If we were to get into a lot of biochemical reactions, remembering the chemistry, the types of reactions that those molecules undergo, which we did not really study, is your key to understanding biochemistry. So a lot of organic chemistry and biochemistry. All right, amino acids with neutral R groups that are either polar or basic. And again, this really shouldn't say R groups. It should say polar acidic and polar basic, because they're certainly not neutral. At physiological pHs, bodily fluids, the carboxylic acid groups will have lost their hydrogen and have the negative charge. Here's histidine, which is very similar to one we looked at just a moment ago, and that nitrogen right there gets the positive charge. These are amine groups, bases. Okay, nitrogen. When we talk about bases, you'll see nitrogen in a large number of bases. So we're going to identify each of the following amino acids as either polar or nonpolar. So the thing to look at is the carbon in the middle. Go back. Carbon in the middle. And one of the hydrogens is that hydrogen that's present on every amino acid, but the other hydrogen is the R group. So this has just a hydrogen. So that is going to make it nonpolar. And then this one has, as its R group, right here is the R group, has an OH group, so it is definitely polar. It's an alcohol. In fact, it's essentially ethanol. And we see that we were correct. Now, we've introduced a word here that, if you were Johnny on the spot and printed notes out before January the 3rd, then I did not have any discussion in the biochemistry section about chirality. I inserted a couple of paragraphs about that. What a chiral molecule is when you have a carbon atom that is surrounded by 4 different groups. So we are looking at a Fischer projection, which is just another way of looking at a molecule. We do it with amino acids, we do it with carbohydrates. In the middle is that carbon. Okay, so these are amino acids. Here's the hydrogen that's present on all. Here is, oh, that's not an amino acid. Here we are. So here's our amino acid. So you have your carbon, and every single one of these at the intersection of these lines. We've talked about line diagrams. Here's the hydrogen that's present on all. Here's the carboxylic acid, and here's the NH3 that's present on all of them. Okay, so the thing that we want to concentrate on is the R group. So it's CH3, that's alanine. Okay, this carbon group in the middle, not this one or this one, but this carbon group in the middle is surrounded by 4 different groups. That makes it chiral. And what happens when you have a chiral molecule is that chiral molecules are able to rotate light, polarized light. And they always come in 2 forms. Form where you can rotate, and the little star is meant to show that's the chiral carbon. In one form, this one, it rotates in one direction, and in this one, it rotates in the other. So those stand for Latin words in terms of the direction. Okay, and I don't remember which is which. But they always come in pairs, and they're called enantiomers. This is covered in the notes. Let me write that word out. I don't know why in PowerPoint I cannot do this well. Enantiomers. That's the word. The 2 forms of a chiral molecule. Then they're mirror images of one another. So if I drew a mirror right here, these 2 are mirror images of one another. This one rotates the polarized light in one direction. This rotates the polarized light in the other direction. In all biochemical systems, only one of the forms is natural. And in the case of amino acid, it's the L isomer. So these are two forms, two molecules that are identical except for how they're arranged in space. Okay, that's called a stereoisomer, how they're arranged in space. And the two forms are called enantiomers. This is further organic chemistry that we did not get into. But I want to talk about it here because it comes up, this idea of chiral comes up when you're talking about L versus D. You might see it as plus versus minus. I go through that in the notes in just a couple of paragraphs. So that's what's going on here, is we're showing that, first of all, that they're molecules that can rotate light. They come in two forms. Only one form is present in nature, either the L or the D. And it's not always the L, but it's always the L when it comes to proteins. You'll see when we get to carbohydrates that it's the D isomer that's used, that is present naturally. Okay, so just showing the two forms of alanine and the two forms of cysteine. In nature, only the L is used. Now, when I take two amino acids, let's say it's glycine and alanine, and I put them together, I'm going to do what is a very familiar reaction in biochemistry, and that is a condensation reaction, or they call it a, when you put hydrogen in, it's called when you put water in, it's called hydrolysis. When you take water out, like we are here, it's called condensation. And there's another word that's used as well for condensation, and we'll come across it in a minute. So what we're doing is we're taking the oxygen, look at the difference, look at this group here, and then look at it right here. So what happened on one amino acid on the carboxylate end is we took the oxygen off. So this oxygen came off. Look at the difference between this amine group and this amine group here. Okay, what happened is two hydrogens were taken off. So this oxygen and two of these hydrogens are forming the water. And it's all color coordinated for you here. So it's a condensation reaction. We're taking water out and you're forming an amide bond. It is a, remember it's an yes, it is like, an amide is like an ester, except the OH is replaced with an amine group, right? Right. So we learned that in our functional groups in lecture 14. It forms between the carboxylate end of one and the amino of another. So this is how you stick amino acids together to form the primary structure of proteins. Of course, there's always going to be a free amine group that's always written on the left, and there's going to be a free carboxylate group, and it's always written on the right. So in proteins, we talk about going from an N to the C terminus for a peptide. So when I start putting amino acids together, it is no longer, they are no longer amino acids. Those negatives and positives go away. Now I have a peptide, and this is a dipeptide because I have two amino acids. Dipeptide. So remember, what's going to happen is I am going to remember to press the pen function. Is this oxygen and two of these hydrogens make the water, and this get stuck together like pop beads to form a peptide bond, which is an amide. An amide group. All right, so two, and this is how we name them. The one that lost the oxygen gets the eel ending, so glycine becomes glycyl, and we leave the other one alone, the one that had the nitrogen amino group used. So the one at the nitrogen terminus gets the eel, and the one at the carboxylate or the carbon terminus, so here's the nitrogen end, because it ends in an amine group, and here's the carbon end because it ends in a C group. And this is true no matter how many amino acids you've got in between. All right. So, eel ending for the N terminal, one that has it's at the left, and then the full name for the other end. So, what if you have three like this? This is a tripeptide. It shouldn't say dipeptide up there, it should say tri. Because you've got alanine, glycine, and serine. So it's alanine, glycine, because they're at the nitrogen end, and then serine ends it with the carboxylate end. So if you did it in terms of the three letter abbreviations, it would be alaglycir. If you did the one symbols, it would be this, which still to this day looks strange to me. So this should say tripeptide, not dipeptide. Guide to drawing a peptide. Draw the structures for each amino acid. Put the N-terminal end of the amino acid on the left. Then you know what to do in the middle. You're going to take the oxygen off the carboxylate, 2 hydrogens off the N-terminal, and shove them together to form the amide bond. Right? And then you connect the remaining parts of the amino acids. So no matter how many you have, that's how you're going to do it. So we're going to take these 2 amino acids, which I fixed this and it's still a little off. This bond here should be to this carbon, so let's just kind of do that, all right? So we're going to take these 2 amino acids and put them together, serine and threonine, all right? So remember, this oxygen comes off and 2 of these hydrogens come off. We put them together, all right? And the dipeptide would be written like this if you were doing the 3-letter abbreviations, and this would be serial threonine. No change on the right, on the amino acid all the way to the right. So peptide bond, amide bond, there it is. There's our amide bond. Whoop. Here's our amide bond right here. Here's our serial. Here's our threonine. It's done correctly here. I can't tell you that how much formatting I had to do to get this, their original PowerPoint to work for us. Condensation reaction. Right? So write the names and 3-letter abbreviations of the tripeptides that could be formed from 2 glycines and 1 alanine. So essentially, how many different tripeptides can you form from 2 glycines and 1 alanine? All right. So how many different forms are there? I know in math there's a way of figuring that out, but we're going to do it. So we could do GLY, ALA. That'd be 1. We could put the glycines on the ends, gly, ALA, gly. Or we could do the ALA first, followed by the 2 glys. And that would be the 3 different tripeptides that can be formed from those 3 amino acids. All right. So if I change it up, now I have 3 different amino acids, okay? Then it would be more, okay? When I had 2 glycines and an alanine, it was only 3 different ones, and now it is 6. So before I had 3, but 2 were the same, and I formed 3. I had 3 amino acids, but 2 were the same. I formed 3 different tripeptides. This time I have 3 different, and I'm going to tell you that it's 6. So why don't you give that a try before I either pause the lecture right here or before I switch to the, and I'm not going to write it out in my horrible handwriting on this PowerPoint, but you can go ahead and give it a try. And there are the 6 different tripeptides that you could form. It would be, it's leucine, so it would be leucyl, glycyl, alanine, leucyl, allyl, glycine, allyl, leucyl, glycine, allyl, glycyl, leucine, glycyl, allyl, leucine, glycyl, leucyl, alanine, if I were naming them. All right, so write the name and 3-letter abbreviation for the following tetrapeptide. So it is 4. So what we're going to have to do is you're probably going to have to, and I tried to fix this and I couldn't. So just a little just a little clarification. That carbon, that's bonded, that's good, that's good, that's good, that's okay. Those are bonded together. That's there, it's good, good, good. So that's the only one that's really bad is this one right here. All right, so my R group is a methyl group. All right, so the methyl group amino acid is alanine. I just happen to remember that one. All right, so that's the 3-letter abbreviation. So it would be allele. Here is an amino acid with this R group, and I am not going to remember what that is. This one I know, it's cysteine. And this one I'm going to have to look up as well. So you do the same. Pause the video, look up these R groups so you can identify which amino acids these are, and then we're going to put them together, 4 different amino acids, and the name. All right, pause it and give it a try. And then here's the answer. Alanine, it's leucine, cysteine, methionine. I remember with the other sulfur containing. So here's the name. Allele, alanyl, leucine, cysteine, methionine. So the eel ending on everything to the left and the one on the right gets the name is not changed. All right, primary structure of proteins is what we've just been doing. We're taking amino acids and we're putting them together into a chain. This particular sequence of amino acids is the backbone of a peptide chain, and this is the primary structure. When our DNA is expressed through transcription, that is what we're doing. That is what your RNA and all the different processes that have to occur. If you remember your biology, which is biochemistry, when it reads off, those codons, you are forming the primary structure of a protein, and it may be coding for a hormone. It may be coding for what, who knows what, since our DNA codes for pretty much everything that is needed in our bodies. All right. Primary structure of insulin was the first protein to have its primary structure determined, and it goes from the N-terminus from the N-terminus to the C, N to the C, and this protein has 2 polypeptide chains that are linked by a disulfide bond. And notice the amino acid that is involved in that disulfide bond. It is cysteine that has the sulfur group. See, all the disulfide bonds are formed by cysteines. And these are very important structure determining type of interaction. Very, very important. This is disulfide bonding. When somebody gets a perm, you are, I believe it is, forming disulfide bonds. Sure stinks like sulfur. Between lengths of hair. Hair is protein, and so you kink it up. The A chain has 21 amino acids and the B chain has 30. So it's pretty small. Insulin, very important protein, regulating fat storage and blood sugar and all of that. Very, very important protein in our bodies. Very first one to be, for its primary structure to be determined. Secondary structure is what happens when the primary structure interacts with each other. And the first type that we're going to talk about is an alpha helix. And that's going to be easy to remember because it looks like a helix. The secondary structure describes when they form hydrogen bonds. So it's hydrogen bonding. This is important. Secondary structure is determined by hydrogen bonds within a single polypeptide chain or between polypeptide chains. So in that previous slide where insulin had 2 chains, but it was a disulfide bond between 2 of them, that was not an alpha helix because it was non-hydrogen bonding. It certainly didn't look like an alpha helix. It has a coiled shape held in place by hydrogen bonds between the amide groups and the carbonyl groups of the amino acids along the backbone. You'll see it in a moment. Hydrogen bonds between the H of the NH group and the O. Okay? And it's specific because of the size they are and everything. So if I look here, it's the carbonyl group on the backbone of the We call the primary structure the backbone. So they have carbonyl groups. So the carbonyl group on this one and the NH group on this one, they are forming hydrogen bonds between them. And this forms, and if it has, forms an alpha helix. Okay, so it goes from C to N. Right. Another type of secondary structure is a beta pleated sheet, and it is formed when you have adjacent chains. When the rigid structure of the amino acid proline causes a bend. Proline was that weird one with the chain. Okay, remember the little cyclic group. So go back and to a previous slide and look at your structure for proline. But it had carbon, and then it had it looked like this. And on the nitrogen was of the amino part was part of it, and it formed like a chain. And there were other groups here. So not a chain, a ring. We have hydrogen bonds between the chains, so you have to have the proline, and the R groups are above and below the sheet, and is very typical of fibrous proteins such as silk. And this is what it looks like. So here you have those hydrogen bonds again. So both alpha helixes and beta pleated sheets have hydrogen bonds between the backbones, but the beta pleated sheets has the amino acid proline in a certain place to cause this type of structure. Another type of the last and third type is a triple helix, and it consists of 3 alpha helixes woven together. It contains large amounts of glycine, proline, hydroxyproline, and hydroxyl lysine that contain lots of OH groups for hydrogen bonding. I should fix that. That's not another bullet. It is found in collagen, connective skin tissue, skin, tendons, and cartilage. So I thought this picture here was rather interesting. I'm assuming that in a young person, and that triple helix is held together much better, maybe, although that's not the way it's depicted here. And in an older person, those connective tissues, that collagen starts to break down. So here is proline. There's that structure we were talking about, but it has an extra, it's hydroxyproline, so it has an extra OH group on it. Right here, hydroxyproline. So here's that molecule, that ring that is responsible for the beta pleated sheets. All right. Lysine normally has 4 carbon atoms ending with an amine group. In the middle of those carbon atoms, we have an extra hydroxy group in the case of hydroxylysine or hydroxyl lysine. Right, so indicate the type of protein structure as either primary, alpha helix, beta, sheets, or triple helix for each of these. So polypeptide chains held side by side, hydrogen bonds. Let's go through them and see if we can't, do some elimination. Corkscrew shape with hydrogen bonds. I think that pretty much tells us that we are talking about an alpha helix. All right, so alpha helix is C. So let's put a 2 there. Sequence of amino acids in a polypeptide chain. That would be our primary structure. 3 peptide chains woven like a rope is our triple helix. So held side by side is going to be our beta pleated sheet. All right. Here we go. Tertiary structure. So we have primary structure, which is the sequence of amino acids. Another bullet I need to fix. We have our secondary structure, which is interactions between the chains of primary structure. And then we have tertiary structure, which gives us the 3-dimensional shape. So it involves attractions and repulsions of the R groups. It is stabilized by hydrophobic and hydrophilic interactions, salt bridges, hydrogen bonds, and disulfide bonds. So you have a lot of things going on here that gives us the overall shape of a protein, the tertiary structure. So here is a protein. It doesn't look like it's been identified. The interaction that the R groups give is three-dimensional structure. So any given protein may have um, alpha helices. It may have some beta pleated sheets here. Here we have some uh, sheets, but they're not pleated so much. Um, and you've got interactions and between um, in this deal here, um, this spaghetti looking uh, stuff is essentially just protein backbone, just primary structure. So this is, no specific secondary structure here, but it's still amino acids all along here. So that's what you're seeing here with the CH3 group coming off this alpha helix and a CH3 group between on an amino acid here. That would be alanine. They're going to be mildly attracted to one another. Same thing here. Van der Waals interactions, London dispersion forces between these nonpolar groups. But you've got hydrogen bonding here. You've got salt bridge between this plus and this minus. That's called a salt bridge because the salt is an ionic compound. That's what we call salts in chemistry. Hydrogen bonding and so on. So the tertiary structure can be, a number of different things going on, both primary structure and secondary structure interacting together to give a specific three-dimensional tertiary structure. And this is very important when it comes to enzymes. The active site of an enzyme where the enzyme does its work may very well be nestled in between all of this. So it's very, very specific and so on, and very, very important. So, summary of the types of interactions in tertiary structure. If you're nonpolar and nonpolar, you don't like water, so you have a hydrophobia. If you're polar and neutral, you're hydrophilic. If you've got some plus and some, if you have plus and minus going on, then you're a salt bridge. If you're neutral and neutral, such as this, and you've got hydrogen bonding, and then if you've got sulfur, these SH groups from cysteine, you can form disulfide bonds. I don't remember if that previous protein, yes, it did have some disulfide bonds right here. Oops. Wait, went too far. Stop it. This one. So I have some disulfide bonds right here. Those are between cysteines. All right, globular proteins, a glob, looks like a glob, have compact spherical shapes. They carry out synthesis, transport, and metabolism. Myoglobin is a globular protein, and it stores and transports oxygen. So I believe this is a picture of globulin, myoglobin, and it has a heme group, which is right here. And heme groups bond oxygen, and they transport oxygen in your blood and in your muscle, and you need myoglobin to do that. So there's that heme group. This is where the oxygen bonds to that to the iron. You've got a carbon end and a nitrogen end. So this is one end of the protein, that's the other end of the protein. Then you've got all those secondary structure interactions, tertiary as well, to bring it into form. Fibrous proteins are long fiber-like shapes. It makes sense that hair and wool, skin and nails, feathers contain lots of alpha helices. Things such as feathers contain beta keratins, large amounts of beta pleated sheet structures. All right, so in the initial form of this slide, it did not give you the R groups for the two amino acids, so it's asking you to select the type of tertiary interaction. Is it going to be disulfide? Is it going to be ionic, hydrogen bonds, or hydrophobic? So I put in the R groups for leucine and valine. If I look at this, it tells me all carbons and hydrogens. So This is going to be number 4, hydrophobic. No, not going to like water. Alkane-like. 2 cysteines. Okay, got that SH, so it's going to be number 1, disulfide bonds. We've got aspartic acid with a negative on its carboxylic acid group, and we've got lysine with a positive on its amine group at the end. So we're going to have some ionic salt bridge interaction here, and then serine and threonine. You've got OH on your serine and OH on your threonine. So you're going to have some hydrogen bonding going on with those. All right. Now, one other type of structure is when you take quaternary structure. You got primary, secondary, tertiary, quaternary is the combination of 2 or more polypeptide chains. It is stabilized by the same interactions found in tertiary structures. Hemoglobin, this is the protein hemoglobin. It has essentially 4 components, and it's shown here with the colors. You've got, and every component has a heme group. So you can do 4 oxygen molecules can be bound to these heme groups. So you've got 2 alpha chains and 2 beta chains with heme groups in each subunit that picks up oxygen. So you've got 2 alpha chains and 2 beta chains. So here we've got I don't know which is which, but you've got this is 1, 2, 3, 4. And each one of these has in the subunit, so we got 4 subunits, has a heme group where the oxygen can bind. Very important protein and found in our red blood cells, needed for transporting oxygen. So, summary of protein structures. Primary, peptide bonds joining amino acids into a chain. Secondary, alpha helices, beta sheets, beta helices as well, and triple helices formed by hydrogen bonding between the atoms in the peptide bonds. Tertiary is when you fold into a compact three-dimensional state stabilized by interactions of the R groups. So secondary is hydrogen bonding, not between the R groups, but between, remember, the carboxylate group and the nitrogen group on the chain on this primary structure. Tertiary is interactions between the R groups. And quaternary is when you take subunits and you put them together to form a biologically active protein. Not every protein has quaternary structure. All right, so not every protein, you have to take protein subunits and put them together, but many do. See, because a tertiary can be biologically active as well. All right, so quaternary is when you take more than one subunit, protein subunit. You put it together to form a biologically active molecule. These protein subunits by themselves would not be biologically active. So in pictures, primary structure, amino acids joined together through peptide bonds. The interaction between these OH groups on the backbone and these NH groups on the backbone, not on the same, not next together like that, but when they come close together, form the secondary structure. So hydrogen bonding, I should do here, hydrogen bonding between the COs and the NHs on the backbone. That makes secondary structure. The interaction of R groups, R group interactions, is what gives rise to tertiary structure. And then the same types of things happen for quaternary structure. So probably a bit of all of it. So R group and probably more hydrogen bonding gives rise to quaternary structure. All right, and hydrogen bonding. All right, so identify the level of protein structure. 1, primary, 2, secondary, 3, tertiary, 4, quaternary. So a beta pleated sheet, is it primary, secondary, tertiary, or quaternary? It is secondary. Right? Once you try these, stop the video and try them yourself. Right? and then come back to get the answers. So A is secondary. Order of amino acids in a protein, primary. A protein with 2 or more peptide chains. I'm going to go with secondary structure there. Might be wrong. The shape of a globular protein. I'm going to go with 3. Disulfide bonds between R groups. Definitely 3. Now we're missing 4, so perhaps the shape is not 3 but 4. So let's check our answers. Oh, 2 or more peptide chains is 4 is the quaternary structure. And I had said 3 and 3 here. All right. All right. So a protein with 2 or more peptides is a little misleading. I would say maybe 2 or more peptide subunits would make it a little more clear. All right, that brings us to the end of peptides. Of proteins. And now we're going to start carbohydrates. I'm going to get out of the PowerPoint for just a moment. Oh, we got 50 minutes, so this is a perfect time to stop section A, and we're going to then start up part B of lecture 17 with carbohydrates.